Experiment 4. Gas absorption desorption is part of the coursework for B49CA, Separation Processes A. In the absorption experiment, air and CO2 are mixed before being fed into the bottom of a packed column, while water enters the top of the column through a centrifugal pump. Counter current flows along the packing increases the transfer of CO2 from the air mixture into the water. The lean air mixture exits at the top, whereas the CO2-rich water accumulates at the bottom of the column and overflows out into a receiving vessel. The opposite of absorption is called desorption. CO2, now present in the water, can be stripped or dissolved into clean air in another packed desorption column. The CO2-rich water is heated before entering the column to decrease the solubility of CO2 in water. The clean air that is fed to the bottom of the desorption column flows countercurrently among the packings with the liquid, causing CO2 to transfer from water into the air. Heat from the water emerging at the bottom of the column is recovered in the coil heat exchanger. It is further cooled by cooling water before overflowing into a receiving vessel. The objectives of the experiments are to demonstrate the absorption of CO2 into water using a packed column, to calculate the rate of absorption of CO2 into water, to determine the saturation composition of CO2 in water, to study the effect of gas flow rate, gas inlet composition, and liquid flow rate on the rate of CO2 absorption. Figure 1 shows a gas absorption desorption unit. The unit consists of a condenser W1, which is a stainless steel water-cooled coil condenser with 0.2 square meter heat transfer area. There is also an absorption column K1 and desorption column K2. These are packed columns filled with 10 mm glass ratchet rings to 1.5 meter effective packing height. Both columns are 80 mm in diameter, and they are made of borosilicate glass. There is also a liquid preheater W2. This unit is located behind the control panel. This unit is a stainless steel temperature control immersion cartridge heater with safety level switch. The liquid preheater has 4 kilowatts heating power. Next is the heat exchanger W3. This unit is a stainless steel coil heat exchanger with 0.2 square meter heat transfer area. Next are some tanks B1 and B2. These are stainless steel rectangular tanks with removable top cover and level side tube. Each tank has a maximum capacity of 50 liter and comes with a low level switch for protection of the centrifugal pump from dry run. Lastly, there are circulation pumps P1 and P2. These units are polypropylene magnetic drive seal-less centrifugal pumps. Each pump is rated at 60 liters per minute maximum delivery, 5.6 meter maximum heat, 65 watts output power. For this experiment, however, take note that each pump is set to deliver a maximum flow rate of 4 liters per minute of liquid only. This video shows general startup procedure. First, ensure all valves are closed except the ventilation valves V14 and V16. Check that all gas connections are properly fitted. Switch on the air compressor P3 and allow the pressure to build until 7 bar. Open the air compressor valve. Set the supply pressure to between 4 and 5 bar by turning the air filter regulator knob clockwise. Open the valve on CO2 gas supply Set the supply pressure to between 1 and 2 bar by turning the gas regulator knob clockwise. Turn on the power for control panel. Check that all sensors and indicators are functioning properly. Allow cooling water to flow through the condenser W1 and heat exchanger W3 by opening valve 18 and valve 19. This unit is now ready for experiments. Experiment 1. Absorption of carbon dioxide from air into water. Before we start the experiment, perform the startup procedure as described before. Step 1. Fill the sump tank B1 with 30 liters of deionized water. Open valve V4. Fill the bottom overflow tube of absorption column K1 with liquid as follows. Switch on the circulation pump P1 and slowly open valve V10. Observe liquid entering the top of the absorption column. Flow down the column and accumulate at the bottom until it overflows back into the sump tank B1. Switch off the circulation pump P1 and close valve V10. Introduce air and CO2 mixture into the absorption column K1 as follows. Determine the appropriate air and CO2 gas flow rate in order to achieve the desired inlet gas composition for the absorption process. Open and adjust valve V2 to set the flow rate of air. Open and adjust valve V1 to set the flow rate of CO2 gas. Prepare the unit for CO2 composition measurement from absorption column K1. Close valve V17. Open valves V15 and V16 and slightly close valve V14. Record the initial CO2 composition as the feed composition value. Introduce liquid into the absorption column K1 as follows. Switch on the circulation pump P1 and slowly open valve V10. Observe liquid entering the top of the absorption column. Flow down the column and accumulate at the bottom until it overflows back into the sump tank B1. Adjust valve V14 to fix the desired inlet liquid flow rate. At regular intervals of 5 minutes, collect a sample from the outlet liquid of the absorption column K1. Analyze the collected sample to determine the composition of dissolved CO2. Continue to record data, collect liquid samples until the CO2 composition in the outlet gas has reached the same value as the feed composition. Repeat the experiment by changing the gas flow rate, gas inlet composition, and liquid flow rate. End the experiment by performing the general shutdown procedure as described in the lab manual. Experiment 2. Desorption of carbon dioxide from water into air. Perform the startup procedure as described previously. Fill the sump tank B2 with 30 liters of CO2 rich water from experiment 1. Close valve B4 and open valves V6 and V7. Introduce liquid into the desorption column K2 as follows. Switch on the circulation pump P2 and slowly open valve V11. Observe liquid entering the top of the desorption column. Flows down the column K2 and accumulate at the bottom until it overflows back into the sump tank B2. Adjust valve V11 to fix the desired inlet liquid flow rate. Switch on the liquid preheater W1 and set the temperature set point at TIC03. Wait for the temperature to reach its set point value before proceeding. Introduce air into the desorption column K2 by opening and adjusting valve 13 to set the desired flow rate of air. Prepare the unit for CO2 composition measurement from desorption column K2. Close valve V15, open valve V14 and slightly close valve V16. At regular intervals of 5 minutes, collect a sample from the outlet liquid of the desorption column K2. Analyze the collected sample to determine the composition of dissolved CO2. Continue to record data and collect liquid samples until CO2 composition in the outlet gas has dropped to 0%. Repeat the experiment by changing the air flow rate, liquid flow rate, and liquid temperature. End the experiment by performing the shutdown procedure as described in the lab manual. Liquid sampling procedure. Samples can be taken from the liquid entering and exiting both the absorption and desorption columns for analysis. The sampling valves are located at V8 for inlet liquid to absorption column K1, V3 for outlet liquid from absorption column K1, V9 for inlet liquid to desorption column K2, V12 for outlet liquid from desorption column K2. The procedure for collecting a sample are as follows. Drain any stagnated liquid above the sampling valve. Place a conical flask or a beaker below the sampling valve. Open sampling valve and collect about 100 ml of sample. Close the sampling valve. Note that you can also use a beaker instead of a conical flask. Analysis of dissolved CO2 in water. Water used for absorption should be deionized because presence of dissolved salts could affect the analysis method as described below. The chemicals needed: deionized and CO2-free water, ER-grade phenolphthalein indicator, 1 liter of standard 0.05 molar sodium hydroxide solution. The procedure: obtain a sample as described previously. Prepare exactly 100 ml of sample in a conical flask. Add 5 to 10 drops of phenolphthalein indicator. If the sample turns red immediately, no CO2 is present. If the sample remains colorless, titrate with a standard 0.05 molar sodium hydroxide solution. Measure the volume of 0.05 molar sodium hydroxide required to reach the endpoint. The endpoint occurs when a definite pink color persists in the solution for longer than 30 seconds. The next video shows a standard procedure for titration.
The purpose of a titration is to quantitatively determine the concentration of an unknown solution, commonly called the titrand or analyte, by adding a volume of a chemical with a known concentration called the titrant. To mark the endpoint of a titration, an indicator is added to the analyte. Let's review some of the equipment that's needed for a titration. Carolina's complete burette assembly contains the basic equipment to get you started, including a burette, ring stand, and a burette clamp. A burette is a long, narrow, graduated tube used to add titrant. It has a stopcock to regulate the flow of liquid. Notice that the markings on the burette go from lowest at the top to highest at the bottom. The ring stand and burette clamp are used to mount and secure your burette. Other materials you will also need include a small funnel to help you fill the burette, a 125 milliliter Erlenmeyer flask, which works best for titrations. The shape of the flask allows for more vigorous swirling than a beaker or other glassware and minimizes spill hazards. A volumetric pipette and pipette bulb to transfer a known volume of analyte to the flask. A wash bottle filled with deionized water, a beaker or flask of titrant and of analyte, indicator selected for your reaction, a reading card to help read the meniscus, and a sheet of white paper to help visualize the endpoint. To prepare your burette for the titration, it is good laboratory practice to rinse your burette thoroughly with deionized water, then with a small amount of titrant. After each rinse, open the stopcock to allow the liquid to drain out the bottom. If there is a lot of liquid clinging to the walls of the burette, then thoroughly clean your burette and repeat the rinse process. The accuracy of this technique is dependent on the titrant flowing into the flask and not sticking to the walls of the burette. Mount the burette in the clamp, making sure it is positioned vertically and there is enough room to position your flask underneath the tip. Check to see that the stopcock is in the closed position. Insert the funnel into the top of the burette and fill with titrant almost to the top. Filling exactly to the 0 mark is not necessary, but you do need sufficient titrant to complete the reaction. Check the column for air bubbles and gently tap to free them from the side walls. Remove the funnel. Place the Erlenmeyer flask under the tip and open the stopcock to allow a few milliliters of titrant to flow through, releasing any trapped air. Rinse the tip of the burette with water. Empty the liquid into a waste container and thoroughly rinse the flask. It is not necessary to dry the flask. Record your starting volume. It is important to accurately read the volume on the burette. First, note that the liquid forms a concave meniscus because the water pulls itself up the side walls of the glass. You should read the volume from the bottom of the meniscus at eye level. Secondly, it is important to read your volume to the correct number of significant figures. This burette indicates volume to the nearest 0.1 milliliters. With any graduated glassware, record the volume to one additional decimal place beyond the smallest graduation. In this case, record the volume to the nearest hundredth of a milliliter, estimating the final place. Use the volumetric pipette to transfer the analyte to the flask. Add a few drops of indicator to the flask and swirl. Place the flask under the tip of the burette. Record this volume. Placing a piece of white paper under the Erlenmeyer flask may make it easier to detect the color change. Operating the burette requires 2 hands. One hand turns the stopcock, while the other hand swirls the flask. Practice turning the stopcock a few times to familiarize yourself with how quickly the flow starts and stops. It is common laboratory practice that the first titration is an estimate. Open up the stopcock and allow the titrant to enter the flask quickly. Continuously swirl the flask. As the volume of titrant in the flask increases, the color of the indicator appears, then disappears as you swirl. When the analyte becomes a colored solution, close the stopcock. Record the final volume of the burette. Subtract the initial volume from this final volume to get the estimated volume of titrant needed for this titration. Knowing the volume that is a little past the endpoint, subtract 5 milliliters from that number to get the amount of titrant that can be safely added before a slower addition is required. As the estimate titration shows, a dark-colored solution indicates an excess of titrant has been added. The desired endpoint is a pale, faintly colored analyte. To reduce the risk of passing the endpoint, slow down the addition of titrant when flashes of color begin to appear in the analyte. Adjust the stopcock to slow the flow of titrant to a drop-wise rate. Continue to swirl the flask with one hand and keep the other hand ready to close the stopcock. When you notice the indicator taking longer to fade, close the stopcock. Rinse the tip of the burette with deionized water and swirl the flask. Record the final volume on the burette. If the analyte remains faintly colored, then you have reached the endpoint. If the analyte is still colorless, then repeat these steps. Add a drop, rinse, swirl, and record until a faint color persists. Typically, titrations are performed in triplicate. Use the average volume of the titrant required to reach the endpoint for any calculations.
For further information, you can contact me, Dr. Noismiza Yusof, by emailing n.yusof@hw.ac.uk or call me at 03-8894-3702. Or you can also contact Harry Ruziana Rusman, our Senior Laboratory Technician, by emailing her h.rusman@hw.ac.uk or call her at 03-8894-3708.